Welcome you all to this Cryo-EM course. Last class we discussed about different types of purification, and we discussed about the size exclusion chromatography or gel filtration, and from there we are selecting our protein of interest, and then we perform dialysis to remove high salt concentration or glycerol concentration, as well as we perform some SDS page gel or native page to confirm our protein size, and then next step we are ready to go for our TEM study. So, now we will start the STEM study. Before that, let us take a look that how we can do the STEM study. There is a two different thing. I already told that okay, there is a purified sample and there is a cell sample, cell or bacterial sample. So, when there is a purified protein, generally this will be less than nanometer thickness. Okay. And the other one, if we just consider above, so most of the time cells, and this will be our purified protein samples, DNA, RNA, viruses, purified viruses like that. So, the thickness is also very important, because previously we also discussed about that if the thickness, if the sample is very thick, electron beam cannot penetrate. So, for the scanning electron microscope, if sample is thick, there is not a big problem, because we are mainly recording the secondary electrons. What is that secondary electrons? If for the scanning electron microscope, if thickness is very high, so mostly electron will come and scattered. So, we are taking that back scattered electron, or the secondary electron will come, hit and displace their position, that electron will go. So, we are just recording the back scattered electron or the secondary electron for the scanning electron microscope. But for transmission electron microscope, if this is our sample, electron beam is going through this sample. When electron beam is going through this sample, we are imaging. So, electron beam needs to transmit. If it cannot transmit through the sample, if the thickness is very high, it is going to block everything. We cannot see anything at the screen. So, that is the reason thickness is a very important part here. So, in this particular case, thickness should be less. Now, this one we can split into two half here. One is, as we discuss these things multiple time, negative staining, and this one is the plunge fringe. So, this one is the plunge fringing we can consider. Now, when we have this negative staining, what we can do? We can just take the snapshot of any image that we just want to collect some 2D images. That is also possible, no one is stopping us. Here structure is not involved, we are just taking some 2D images, and it may be the protein, DNA protein. So, if there is a protein, I want to see that it is binding to the DNA, and if we can collect some nice images where protein is binding to the DNA or protein is binding to the RNA. So, RNA or protein is interacting with the lipid molecules. So, we can just take some 2D images. So, and then we can tell that okay, I can see very clearly DNA protein interaction, very clear RNA protein interaction images or lipid protein interaction images. I will show you all these example in the later stage, next class, when we will discuss about the good images and bad images. Now, if I come here, now this one sometime, or if you just consider 10-15 years back, lot of time these negative staining images we are using tilt pair reconstruction. So, it is a random conical tilt we are collecting. So, 3D structure we can do very low resolution. Okay. Why negative staining is always giving low resolution images, that we can discuss in the later stage, but most of the time we are using the random conical tilt experiment or orthogonal conical tilt experiment. So, random conical tilt experiment is very popular in 2000 to 2015, before we got the direct electron detector. So, we are using that random conical tilt to get a low resolution structure and just a volume. And the other way that okay, so if I just use this one, other possibilities, you can just get a 2D crystal images. We can get a 2D crystal images and then we can do electron crystallography. So, we can do electron crystallography, right. Now, we are coming to this particular stage, that okay, plunge freezing. So, plunge freezing, mostly what is required in plunge freezing, this is we are using this particular one for single particle analysis. We can tell single particle analysis cryo-EM. So, single particle analysis cryo-EM, what is required? We need a isolated single molecules, single biomolecules, which we can separate it properly. If they overlaps with each other, if they aggregate, the single particle analysis is not going to work. That is why it is telling single particles analysis. So, single molecules are required. So, this plunge freezing method, we can use it single particle analysis for any protein sample, where we have a single particles. Other what we can use it? We can use it helical reconstruction, Sometime this negative staining also we can use it, it will be negative staining also we can use it single particle analysis. So, we can use it for the single particle analysis. So, this is our plunge freezing. What about our next, that when you have a thick sample, how we will deal with a thick sample? Let us take a look with a thick sample. So, thick sample we can split it into two half, mostly high pressure freezing. High pressure freezing is very important here, because we are dealing with the entire cell or bacteria. Inside that cell and bacteria there is a lot of liquid material will be there, and if we use the entire cell or bacteria to freeze, so the inside the cell it will be very slow process, because overall if you look at the bacteria or cell will be this big, and the surface when you will try to freeze that one, surface has a different temperature, inside has a different temperature, and gradually this temperature will go down inside the cell. When it will go down gradually inside the cell, so there will be a lot of crystalline ice. That is the reason most of the time we should use a high pressure freezing when you will try to freeze the sample. Then some kind of section or other things we can also freeze it separately. So, here another step, it is called the freeze substitution. So, if you just consider substitution, okay, this can go this direction also. The next, when after high pressure freezing, next step will be cryo ultramicrotome. So, cryo ultramicrotome is an equipment and we are using cryo ultramicrotome to do the next step. So, here we can use resin and form that resin block. I will come back to this step again when we will discuss about the different types of sample preparation. So, when we have this, we use that ultramicrotome again. So, here ultramicrotome techniques again to do the section. So, when we have both the things, that we have a whole cell and we have a thin section of the cell, we can use this thin section of the cell for tilt series collection for cryo-electron tomography or electron tomography. Now, electron tomography, you can use it for these samples also, that you have a bacteriophage or normal phage, or you have a liposome and protein is embedded and you want to do the tomogram, you can use this also as a tomography sample. I will show you couple of these examples when we will discuss about the data processing and 3D reconstruction of this tomography sample. So, here we can collect this tilt series and finally, we can use this for electron tomo. Okay. So, this is the step we can use it to tilt series and electron tomography. Now, let us go back that okay, these are the normal pathway to get different kind of sample preparation, and based on the sample thickness and our requirement we can decide what we are going to use it. So, we can, if the sample thickness is not that big, that if we are not dealing with a bacterial cell or normal cell, most of the time we are dealing with a purified protein sample, then we can go through this pathway, and if we are dealing with a thick sample, then we can go through this tomography pathway. Now, we are constantly talking about the grids, that okay, grids are very important for sample insertion or making that sample. So, what are grids? Why we are talking about that grids? So, if you remember your sample holder, when we discuss about the sample holder, what do we have there in this sample holder? You have a adapter and then you have a rod, there you have a pin which will recognize, then there will be a area, there will be a O-ring here which will directly connect to the microscope, and this is the place where your grid will go. So, this is the entire structure of your holder. So, when we discuss about the sample holder, I also discuss about how to insert that sample and where your grid will go. I will discuss little bit about the grid also that time. So, your grid is a small disk. So, generally this grid will be a small disc of copper, this outside layer it will be a copper layer, right, and then inside there will be a lot of this copper bar and it will form a square. I discussed these things little bit about that what is the difference between the cryo grid and the carbon coated grid. So, now here whatever I draw, I told later stage I will discuss about that grid. So, here you can see there are some examples of grids. So, this is the outer layer of that grid if you just consider, and those are the metal bar. You can see it how this metal bar, and if you just look at this, this is the empty place, nothing is there. On top of that you have to put the carbon, but now you can see that here this is a snapshot of different types of grid taken from the wave. So, here you can see the all these squares are same size, but they are different color. One is little bit brown color, this is a copper grid, this is a gold grid, and this is a nickel grid. So, generally different types of grids are possible, different metal we are using. Also, if you look at the shape of this grid is different from this particular grid. Shape of this grid is different from this one, shape of this grid is different from this. This is just a multiple grids, and then not only that, this particular grid and this particular grid, they are not same. This particular grid is not same. So, based on our requirement, what kind of sample we are imaging, we can use a different types of grids, and that is very important. First we have to select our proper grid when we will prepare our sample. Now, here I am giving some examples. So, there are some of these companies, those grids are these images are taken from some of this website, that there is the grid generally supplied by SPI or Electron Microscopy Sciences, that EMS or Sigma, Labtech, they are supplying the grids. Here also you can see that grid. As I mentioned previously, the diameter of this all the grids, it will be 3.05 millimeter, because if you just go to this one, this diameter is very important because holder we cannot change again and again. So, this is, it can fit only this diameter grids. That is the reason most of the time those grids are fixed. There is a other size is also possible, we are not discussing, we are just discussing the commonly used grid. Now, you can see in these grids all are having same diameter, but there are some differences. What kind of differences are there? Anyone can see those grids and then they can tell that the major differences. The major differences are here. If you look this one, so you can see the square size is different and numbers of squares are also different. So, this is here in this you can see numbers of squares are lesser than this one, numbers of squares are much higher of this one. Now, here, so this is basically telling how many squares are there. Based on that we can tell that what is the mesh size. So, it is a 50 mesh grid or 100 mesh grid or 75 or 200. So, generally 200, 300, 400 mesh grid we are considering. So, here you can see if we just go to this one, you can see this is a 400 mesh grid, where you can see individual squares are extremely small, but the numbers of squares are much higher. So, this one is a 300 mesh grid because total 300 squares are there. The size of the squares are smaller, but you have more squares. So, that will vary that what kind of grids do you want, that do you want a only 50 mesh grid or 400 mesh grid. So, generally for biological science sample purposes we are using 200 mesh, 300 mesh, 400 mesh grid. Generally we are not considering this kind of grid because this the entire square area is so big and overall carbon layer will be very unstable, and here so square size area of the squares is smaller, and that is the reason it is much easier to control this kinds of grid, and that is the reason we are using these types of grid for biological research purposes. Now the next one is the you will get a different types of another different types of grid also, and these types of grid where you have certain kind of marking. So those are called the finder grid, mainly for the cryo-electron tomography. We are using this kind of finder grids because sometime we are using correlative light and electron microscopy technique where you are having a fluorescent tag in a bacterial cell, and the bacterial cell might stay here. In the fluorescence microscope we can see that, and now we are inserting the same grid. So if there is a finder that okay, so my bacteria is somewhere in between F and G, or if I can find out those, if there is some kind of marking, it is easy to find those bacteria. Most of the time these finder grids we are using for the tomography sample, CLEM and cryo-CLEM type of sample where you have the fluorescence tag cell or bacteria. We did the sectioning or using the FIB-SEM sectioning, and same grid we are going for tomography data collection in 300 kb microscope. That time we use this kind of finder grids. Now different shape is also possible. You can see different types of grids are here. So this is the normal empty grids, there is no coating, nothing. In the next class we will discuss about how to coat those grids, and then what is the difference between carbon coated grid and the quantifoil cryo grids. So I will finish today's topic here. So most of this grid, carbon coated grid and everything commercially available, but you can also prepare this kind of carbon coated grid in your laboratory also, that we are going to discuss in the next class. Thank you all for your attention.